The next issue is that we will have the next webinar in 3 months from now. So it would be June 27th, exactly at the same time. It would be Thursday, 10 a.m. Eastern time. So we will kind of keep on having these webinars together. So if there are things that we might not be able to cover for today, we basically have time to discuss about those things in the future kind of webinars. And we are hopeful after this webinar, we might be able to move forward and think about how we might be able to have something like a consensus paper in terms of the method that we are interested to discuss and the quality controls and the standards we like to push forward. These are the things that we will discuss during the meeting today. So just to be kind of on time, I would like to ask others who are already kind of online, and we already have 32 participants from across the world. So I would like to ask people, probably we cannot ask all the people, but we have something like 10 minutes to ask people to introduce themselves. We already have kind of something like 10 different speakers. So our speakers probably can introduce themselves when they want to start to talk about their topic, but others, it would be nice if they introduce themselves, just kind of letting us to know what are the people that we have from different parts of the world. So I have a kind of a list of the people, so I can start from the top of the list and asking people to introduce themselves if they like, and I move forward for the next people in the list. So Amy Lowe, are you online? Okay. Anand Shinde.
Hi, I'm Anand. So I'm postdoc with Dr. Gottfried Schlaug in Beth Israel Deaconess Medical Center and Harvard Medical School. And we are working on brain stimulation and trying to understand different motor functions. And we are trying to understand different levels of brain stimulation in TDCS, different current levels. And we are doing that inside the MRI scanner. We are doing simultaneous stimulation and MRI.
Good.
That would be brief intro.
Great. Kaveh.
Hi, I'm Kaveh Shanai. I'm an electrical and computer engineer, and I'm here, I guess, more for my ability to create those shocks that you guys want to use. And I'm also trying to learn about the brain.
Good, good. Asif. We cannot hear you, Asif. You need to turn on your.
Yeah, okay. Thank you. Hi, I'm Asif. I'm from IFARO. I'm in the group of Michael Nietzsche, here with also Fatime, and we're both postdocs in Michael's group, and we're doing a lot of basic work on TDCS. And in the next year, we hope also to now expand our work to fMRI. We're getting a new fMRI scanner, so hopefully we get some good results.
Good.
Yeah.
Good. Fatima, do you want to introduce yourself?
Yeah, I think the same. Just I think Asif introduced me too. So my name is Fatima, and other information is the same.
Good, good. And then Ben Thompson.
I am Ben Thompson from University of Waterloo in Canada, and I'm interested in using brain stimulation over visual cortex.
Good. Benedict Zoffel.
And can you hear me?
Yes.
Hi, Benedict here. So I'm at the University of Cambridge, interested in neural oscillations and in particular how they relate to speech perception. So we are often using TACS to manipulate these oscillations, and then we test how this affects speech perception comprehension. So the reason why we are interested in fMRI is because it's when we manipulate speech perception using TACS, it's very often hard to say if we are actually addressing a speech-specific process or more acoustic process. So fMRI is very useful because then we can actually test where our effects are. So we've we've run one fMRI TSES study, and we are planning to do more. So I'm very interested in this conference.
Good. Carlo Cavalieri. Carlo, could you hear us? Now, Catherine Stoodley.
Hi, I'm Catherine Stoodley. We've been doing TDCS fMRI for a couple of years. My favorite part of the brain is the cerebellum, because it's the best part of the brain. And so we are interested in using TDCS to understand the contribution that the cerebellum makes to cognition, cognitive development, and neurodevelopmental disorders.
Good. Then we will have Jenny Crinon.
Hi there, from University College London. Thanks for inviting me. My group specialises in brain and language functioning after stroke primarily. So we're interested in concurrent TDCS, fMRI for speech production, but now we're looking at general executive functioning because obviously the frontal lobes does lots of jobs. So thanks for including me. Thanks.
Good. Now, Johannes Wosko.
Hi, can you hear me?
Yes.
Yeah, okay. I'm Johannes Voskul. I am working in Christoph Hermann's lab in Oldenburg in Germany. We are basically interested in brain oscillations and their role in attention and perceptual processes. We've also done one TACS fMRI study, and right now we're also interested in combining TACS, especially with MR spectroscopy.
Good. George Almedia.
Hi, can you hear me? I'm sure you can. Cool. So my name is George Almeida. I am at the University of Coimbra. We are using TDCS mainly as a causal tool to get at questions of object recognition, category specificity, the organization of information in the brain, and we're doing it in tandem with fMRI. A couple of projects, with a couple of papers out on that, and a couple of projects starting. And we also have, and perhaps Catherine could be interested in this, we also have work on aging and cerebellum that she might be very interested in, with very nice results, both behaviorally and neurally also. Anyway, thank you for inviting me.
Good. And then Zhuhang Zhu.
Hi, everyone. I'm Jun Hong, working at Harvard with Amy Low and Brad Nanner. Our group majorly focuses on the application of TDCS on older end of population, about the cognitive and motor function, like balance and mobility and dual task control of the balance. And we are we're focusing on some like people, like population with some dysfunction, like Parkinson's disease, and those with slow gait and impaired cognitive function. Also, we are doing some like functional MRI studies to check the effects of TDCS on the brain's network.
Good. Polly Frango. Hi, Polly. We cannot hear you.
Hello, my name is Polly. I'm from the University of Cambridge. I'm a postdoc with Professor Zoe Kurzi, and we use TDCS for visual learning, and we're currently running our first TDCS fMRI and spectroscopy study.
Good. And Zhao Zhang.
Is it me?
Yes.
Okay, my name is Xiaochu Zhang, and I come from China and from the University of Science and Technology of China. And I'm working on the TDCS, and it's the treatment on the nicotine addiction. We published one paper in the social cognitive affective neuroscience in 2017, and we found we used fMRI and to try to find what the neural basis, neural change when we use the TDCS on the smoker and reduce their craving. And this is one paper. Now we are working on the how to use the fMRI to try to predict the effect of the TDCS. Okay, so that's all.
Good, good. Thank you. So we have other kind of people who are basically speakers, or they will have kind of opportunities to introduce themselves in the next steps. Let's move forward to the first kind of short talk. Let me just share my screen. Okay. So, could you see my screen well? Good.
Yes.
Good. Good.
Yeah.
So what I'm going to talk about is just kind of giving you an idea about what we already have in the field in terms of the publications and potentials for kind of moving forward and think about how we might be able to collaborate in the future. So, okay. So we have recently kind of finalized a systematic review, seeing what are the publications that we have in the field with a combination of TES and fMRI. And by the end of January, we have 139 original publications in combination between TES and fMRI. And a group of colleagues basically collaborated on that specific systematic review. We have Peyman, Fateme, Asif, Zainab, people from different labs that basically helped us to do this systematic review. And among these 139 papers, you can see that they are basically, most of them are published in recent years. And we can see that the field is basically growing up, and we potentially see an extensive publication in the coming years. But there are challenges for those publications. So we are going to discuss about those things as well. In terms of kind of dividing those publications in major categories, most of those publications are basically focused to have an kind of a mechanistic idea about how TES work. So they basically do brain imaging most of the time before and after transcranial electrical stimulation, and they try to see what are the mechanistic kind of things that they can find from doing imaging along with TES. But we also have studies that they try to do sort of prediction, trying to predict response to TDCS. And there are studies that they try to use fMRI as a predictor in response to TDCS. And there are studies that they try to consider fMRI as something like the response to TDCS and try to kind of predict what are the things that could be predictors of neural response to TDCS. And meanwhile, we also have studies that they basically use fMRI mainly to just define a specific montage or electrode positioning, and then doing TDCS based on that montage. So most of them are technically studies that they try to do something like causal inferencing based on doing a TDCS on a specific locations that are pre-specified by fMRI. So these are basically three major types of studies that we have. And if I go back to the kind of the Prisma flow chart that we had before, something like 121 of the studies that we found are basically mechanistic studies. And we have 9 prediction studies and 14 montage studies. So you can see that most of the people are basically interested in the mechanistic studies, but there are also potentials in other types of studies as well. And then when you start to dig into those studies in terms of the methods that they are using, whether they basically use post kind of stimulation scanning or pre and post stimulation scanning, or online fMRI, or pre and online fMRI, and online and post fMRI, or pre fMRI, online stimulation fMRI, and then post. So you can see that people are using different sorts of kind of sequences in terms of combining TDCS or TES or TACS with fMRI. And when you go into the depths of the studies in terms of their design, they have different sorts of designs. We have open label designs, we have parallel design, we have crossover designs. And then thinking about, let's say, different sorts of control that they are using, different sort of sessions, how kind of they follow up the results, you can see that there are different variations among. And then we add basically a stimulation protocols, imaging protocols, and how they basically measure the outcome, you can see that we have a huge parameter space that people are exploring, and it's really hard to find two single studies basically replicating what they have been doing before. So it's really kind of a wide range of parameter space that people are exploring, and it's still people are we are all in the kind of exploration phase, but we might need to think about how we might be able to do things in a more coherent way that we can put them together in the future and see how we might be able to make conclusions in the future. So it seems that there are still main things that we need to do to be able to kind of have a more kind of synchronized or coherent activities in the field to be able to have a collective conclusion, even for really simplistic things. And potentially in the next webinar, a few of my colleagues would present you how divergent methodologies that people are using in the field. This is really hard to kind of find any sort of replication or having any sort of kind of meta-analytic conclusion from the publications that we have in the field. And that basically raised a serious concern in terms of how we might be able to increase synchrony in the field in a way that we can make a conclusion or replication, or even kind of something like a collective interpretation from the results that we have in the field. So that's kind of the major concern. And then we are trying to see how we might be able to extend collaborations to fill this gap in the future. That's the initial idea of having the international network of doing TES and MRI together. So that's the initial idea. Okay, so the next speaker we will have, right now we have 7 speakers from different labs across the world that they are basically going to share what they are doing in terms of different methodologies in different labs. And then we will extend that discussion in terms of how we might be able to do something together, or even have something like a shared quality standards in doing these studies in the future, or at least reporting those studies in the future, that would help us to collaborate or think collectively together. So the first speaker would be Lucia Lee from Imperial College, London. The only issue is that I have your slides kind of attached to each other. I can't kind of give you the screen, but that would basically take time if we would just kind of shift the screen. So I will share slides on my screen, and then you can kind of talk on your slides if it is fine. Is it fine, Lucia, with you? Good. So let me just bring up your slides and then share the screen. Okay. Here we go.
Okay, cool, thanks. Hi everyone, it's great to be here, and thank you, Hamid, for convening this webinar. It's really great, and hopefully you'll get lots of good things from it. So my name's Lucia, I'm a clinical fellow at Imperial College London, and the primary things that we're particularly interested in working on is we work with traumatic brain injury patients, partly because that's where our clinical interest lies as neurologists, but also because we feel they are very good models for various syndromes, so executive dysfunction, emotional dyscontrol. They do also have motor problems and apathy. So our focus has been on multimodal imaging even before TDCS or stimulation methods came along. So we're usually doing a combination of structural imaging, fMRI, we also have some PET. We've recently started doing some EEG, though sadly not yet in the scanner, and then computational methods surrounding trying to combine those approaches. As I said before, our key area of focus has been on cognitive function, but I'm sort of moving more towards emotional function in the next few years, and particularly looking at the brain networks which subserve those functions. And then an area of interest particularly for us, is inter-individual variability, partly because it is so relevant for brain stimulation, but it's also relevant for any kind of intervention for traumatic brain injury patients because we have a lot of patient heterogeneity. The stimulation standards that types that we use have primarily been TDCS and TACS, and we've used them both as an interrogation technique. So what can we learn about the brain itself by using these techniques, but also as interrogating them for whether they might be useful therapeutically. And I won't speak too much about our technical setup because Ines is going to talk a lot more about that later. But we've had a lot of input from David Carmichael, who's an MR physicist down at King's, and then David Sharp is the neurologist who's the PI of the lab that I work in. So previous projects that we've published in and looked at, one was particularly looking at the use of TDCS with simultaneous fMRI, and the idea was to interrogate its effect, particularly with regards to the effect of brain state and polarity and that interaction between them. And I guess the sort of particularly novel thing that we tried to do there was to use very short periods of stimulation so that we could get maximum, so we could get both task and rest, and we could get both cathodal sham and anodal stimulation all in the same run to try and increase the power of our analyses. So that's recently come out. It's probably one of your 4 2019 papers, Hamid. So that's recently come out in HBM. And then the next project that we've also been working on is looking more at the use of TDCS as an intervention, and particularly for the stop signal task, which is a marker that we use for cognitive control. And I guess we've done a sort of slightly more traditional approach here in terms of we have a full task during which we apply stimulation, and this is all concurrent. So those 2 papers are in controls, and we've been taking this methodology and taking it forward into patients. That data has been presented at various conferences, most recently in Vancouver, but it's not out yet. So yeah, wish me luck with the reviewers. So I guess can we move on with it? Great. So our emphasis has been on, I would say, simultaneous acquisition. And then when we have, because of that focus on trying to explain variability in response to stimulation, we've also acquired DTI and MRS to try and explain those factors. So our current projects and ongoing and future projects, we have some nice simultaneous TDCS data on resting state, which was acquired through multiband. So one of our collaborators is currently looking at the idea of whether TDCS has criticality. I'm starting some, again, control projects, moving towards patients and looking at the use of TDCS for emotional processing, because our patients are very impaired in that respect. There is Neil Grossman, who has done a lot of work on temporal interference, and we're all trying to find different ways to use his amazing new technology to see whether we can learn a bit more about the brain, but also again for intervention purposes. And then moving forward with the multimodal, whether we can try and combine things like stimulation and EEG, and then stimulation, EEG and MRI. Like I said, I think Ines will be talking a little bit more about the technical aspects, but it'd be great if anybody had wanted to collaborate. We're very open to collaborations, and my email address is there.
Good, thank you, Lucia. Paola?
Me? Yes. Hi, I'm from Rome. I'm the research leader of a research lab, whose research aphasia lab, whose main aim is to find new techniques for the rehabilitation of aphasia for aphasic people. And I'm particularly interested in studying neuroplasticity through TDCS and combining TDCS with other methods such as fMRI. But what I would like to share with you is one method that we used 4 years ago, which is I'm sure you are already aware about this method, the voxel lesion symptom mapping analysis, and I believe this method can be very useful for to study chronic patients, for studying chronic patients, chronic populations, neurological populations, because differently from fMRI, voxel lesion symptom mapping, Sehm can tell us how much each given voxel can be related to a certain task. So it's not that like fMRI is not that we look at all potential regions involved in a task. It identifies regions which specifically are involved in task performance. And I think the one question which still remains to be answered, which is a very difficult answer to be answered, and I don't think that my paper answer to this question, but is we don't really know about the anatomical predictors of TDCS, in the sense that it might be the case, this was our question, that some part of the brain, some areas might be more or less predictive of TDCS recovery. So that's why we use VLSM. We try to correlate, we correlated the performance of the patients before and after ATDCS in three different, we look at the amount of recovery in three different tasks: picture description, noun naming and verb naming, after a training of 2 weeks training, combining ATDCS with a conversational therapy training. And what we found is that we correlated each given voxel with the performance of the patients which were or not, which present or not a damage to that voxel. And what we found is that there were some areas which were more more involved, more responsible for not having find results, in the sense that one particular, we look for the superior longitudinal fasciculus, and what we found that these structures, the patients with a damage to these structures, were the patients who had the worst performance after ATDCS. So this one, I mean, this was the only paper we wrote with VLSM, but I think I would like to share with you this possibility to study together which area might be more or less important to stimulate in order to reach the best recovery after ATDCS. And the other paper, which
Paola,
I mean,
just 1 minute.
Yeah. Yes.
Just 1 minute.
Sorry. Yes, the other method we use is a resistate fMRI. What we usually do, and we are still doing, is to measure the effect of TDCS before and after training, combining TDCS with a language treatment. We measure the impact on functional connectivity through resistate, and I believe that resistate could be a good method for neurological populations, because as you certainly know, some patients are not collaborative. So just asking them to go into the scanner, it's easy, I mean, to measure the possible modifications in the brain before and after TDCS. I think it's, I mean, I don't know if you have any question, and I mean, this is what we are doing.
Thank you, Paola. Now, Bernhard. Bernhard, could you hear us?
Okay, can you hear me?
Yes.
Ah, perfect. Okay. All right. Okay. Thank you very much for inviting me here. Hamid actually came a little bit late because I was not able to calculate the difference between Middle European time and Eastern time. So I made an error for 1 hour. But yes, to shortly introduce myself, my name is Bernhard Sehm. I'm a neurologist. My research group at the MPI in the Department of Neurology is investigating neuroplastic processes that might underlie neurological and sensory motor recovery. And can you please switch to the next slide, Hamid?
Sure.
Okay. Yes. So the idea is to harness plasticity for sensory motor recovery. And just to give you like a short overview of what we are doing, we are using, of course, behavioral measures. Specifically, I will shortly detail that a little bit later, robotics and kinematics newly. And one of our emphasis is to use neuromodulatory techniques such as TDCS and TACS in relation with brain network assessment by functional and structural MRI. The idea is to, yes, of course, to improve behavior by neuromodulation, but at the same time understand mechanisms, underlying mechanisms, for example, by modulating whole brain networks as assessed with resting state functional MRI. And now the next slide, please, Hamid. Okay, so this is just a short overview of previous studies. What we did was first to modulate or to target the primary motor cortex with TDCS and assess resting state functional connectivity changes using both seed-based analysis, but also whole brain graph-based analysis with eigenvector centrality mapping, TDCS setups, so-called bilateral or bihemispheric TDCS versus unihemispheric TDCS. And consequently, we adjusted stimulation parameters and sort of more individualized stimulation parameters in terms of stimulation frequency by using alternating current stimulation, which we assessed first behaviorally in the somatosensory system, and then also using, again, resting state functional MRI to assess changes in whole brain functional connectivity when we apply endogenous alpha or sensory motor alpha frequencies. And here we see like a picture of whole brain changes. We see by adjusting this specific this frequency to endogenous rhythms, we can specifically modulate primary somatosensory cortex, and this is now under review, this result. Please switch to the next slide, Hamid. Another study which is ongoing now is sort of with TDCS now. Here we again compare these two stimulation setups, bihemispheric and unihemispheric TDCS, in stroke patients online during resting state, but also task-based fMRI. We also have two healthy control groups, elderly and young. Outside of the scanner, we investigate behavioral changes with this robotic assessment that I was suggesting before. Here we have quite sensitive assessment of different domains and motor behaviors, including both simple tasks such as visually guided reaching, but also bimanual tasks, and also proprioceptive tasks. We apply, again, TDCS during, as I said, resting state and task, resting state functional MRI, but also task-based functional MRI doing simple movements. So the patients do simple movements by just moving, doing elbow movements and fist movements. And we also acquire structural MRI as potential predictors to predict the TDCS effects. And now please to the last slide. So yeah, this is sort of what we are doing in Leipzig. The first two studies, all three studies that I shortly presented, the data are already there. The last study we are still analyzing. But I would be happy to collaborate in data analysis, for example, with other people here. Sort of, I didn't hear the first introduction by you, Hamed, but so my ideas when I was contacted were it's a very interesting initiative, and I think it would be nice to sort of systematize and compare previous studies that relate TES stimulation techniques on a methodological level, but also on a data analytical level, because I think the ways to analyze resting state functional MRI are very, very variable and difficult, and it would be really worthwhile to find maybe a parameter that across different labs and data sets might be sensitive to, to validly show changes in functional connectivity. So that would be something maybe to discuss, and maybe like in future also, I could imagine also other maybe common projects, including prospective studies. Yes, that's it from my side.
Thank you, Bernhard, for actively contributing to the network. We definitely discuss about those potentials together in terms of how we might be able to move forward. Okay, now Henry from University of Pittsburg.
Hello.
Hello.
Can you hear me?
Yes.
Hello. Great. Thank you. So this is so Hamid asked me to put this slide on. This is our lab. It's run by the PI is Mary Phillips. I'm a junior faculty member. I've been working with her a few years now, and this is our team: Simona, Michelle, Bertocci, Kale, Brian Kaufman, and Ben. We're working with Ben Greenberg, also at Brown. So if you want to go to the next slide. So just a brief introduction. This is how we got interested in this. We're relatively new to TDCS, but there is some work on TDCS, quite a substantial body of work now looking at TDCS as a treatment, potential treatment for mood disorders, particularly unipolar depression. And these trials tend to use the F3, sort of focused on the dorsolateral prefrontal cortex with an F3 anode and F4 or F8 cathodal stimulation. And yeah, as I say, there's some consistent evidence of some efficacy. There have been a couple of recent null result trials, but generally there is a modest evidence of a modest efficacy, probably slightly below that of antidepressants. We were particularly interested in one case report in which an F3 EC extracranial montage actually led to the induction of a hypomanic, or hypomanic in this case, I think, episode. And there have been a few of these studies. A recent meta-analysis suggested that these are relatively infrequent, and obviously transition to mania can occur with antidepressant treatment anyway. So this isn't necessarily evidence of any particular mechanism, but we were interested by the montage that was used in that study, which another study from Colleen Liu's group, Bai and colleagues, suggested that the EC montage is associated with particular current electrical field within subcortical and potentially ventral prefrontal structures. And at the time, we had a number of findings relating the left ventrolateral prefrontal cortex to bipolar disorder. So across a number of studies, we've seen heightened evidence of heightened reward anticipatory activation in bipolar disorder compared to unipolar or controls, also related to potentially bipolar related symptoms such as urgency and impulsivity. So our goal has been over the last few years to see if we can modulate this activation. using or this potential biomarker of bipolar disorder using cathodal TDCS. So if you want to go to the next slide. So this is just a brief overview of what we've been doing. It's a StarStim TDCS system, and we're applying 1 milliamp of cathodal stimulation to F7 with an extracranial anode, and we comparing this with a positive control condition in which we're stimulating at CP1, which is over sort of left somatosensory, approximately cortex, and this is a counterbalance design. So we're bringing participants in, testing them with one montage, and then a week later using the other montage. And our proxy of reward-related activity is obtained from the card guessing paradigm based on that designed by Mauricio Delgado. And we've adapted it to look at anticipatory responses, so monetary anticipation. And that, but just the end, that's a brief, sorry, just, yeah, the last figure just shows the order of stimulation with respect to the different sequences, fMRI sequences.
Good. Thank you, Henry.
Thanks.
Now Ben Thompson, Waterloo.
Thanks, Hamid, and hi everybody. And thanks also, Hamid, for organising this. I think it's a fantastic initiative. In my lab, we're interested in human visual cortex development and plasticity, and we use a variety of different brain stimulation techniques over primary and extrastriate visual cortex and the cerebellum to explore questions relating to vision and eye movement control. And if we could just go to the next slide, Hamid. One of the questions that we've been addressing with combined brain stimulation and MRI relates to a disorder of vision called amblyopia. And amblyopia is a neurodevelopmental disorder that affects vision in one eye, and the loss of vision is due to abnormal processing of information from that eye within the brain. Adult patients with this disorder are typically considered to be untreatable because the brain lacks sufficient neuroplasticity to relearn how to use the eye. And we've been interested in whether brain stimulation can be used as a treatment modality for these patients. So we found some really encouraging results with anode or TDCS over visual cortex, and very recently with random noise stimulation over visual cortex 2. And in parallel, we've been trying to use brain imaging techniques to understand what the mechanisms might be. And the two techniques we've been looking at are functional MRI to look at changes in visual cortex and to look at neurometabolite changes, particularly GABA, which may be related to visual cortex plasticity. You can see me with the electrode array there on the right-hand side. That's what we typically use. One electrode over bilateral occipital poles and the reference electrode over OZ. So if we could just go to the next slide, Hamid. I thought I'd just touch on two of the experimental design considerations that we've kind of come up against in these studies. And I should say, I'm not a technical expert in MRI. I'm a use with the physics experimental designs. The first is how we deliver the brain stimulation. So in all the studies we conducted so far, this is technically more straightforward and requires less kind of development time. But this year's when you start collecting data, and after a lot of practice, we got our data collection down to about 5 minutes. We've done have been up to about 7 minutes. So clearly that's not ideal, and we might use some in-scan stimulation, some online stimulation. That should say 2 milliamps on the slide, not 0.2. Sorry about that. But we haven't been able to variety of technical issues, and so we've gone back to simply doing it outside of the scanner and kind of taken the hit on. Let's go to the next slide, Hamid. The other design consideration that we lot of thinking about is what do you use as a base. So for our fMRI stimulation, we decided to not do a baseline scan, but simply do the stimulation in 2 separate sessions, real anodal stimulation in this experiment and a sham session. And then we compare the ratio activity in visual cortical areas between the 2 eyes. So we cover one eye, provide the stimulation, then cover the other and provide the stimulation. So we kind of have a change in the ratio as our outcome measure, which seemed to work well, and we detected effects. In our study, obviously we get a pre-stimulation scan, then we take them, trying to match the parameters as closely as possible. To hear how other people have addressed this question of baseline, because obviously on what we compare our TDCS effects to. Thank you.
Thank you. Thank you. Thank you, Ben. And now we move forward to Linde. Linde, are you available right now online? Linde, could you hear me? Okay, so let's move forward to the next speaker, Gesa from Germany. Gesa, would you move forward? Can you hear me?
Yeah.
Good.
Can you hear me?
Yes, loud and clear.
So, yeah. Thanks for inviting me. So I'm also from the Max Planck Institute in Leipzig, and my research group focuses on cognition and plasticity, mainly in the language network and in other higher cognitive functions. And I have to say I'm mainly a TMS person, so I only fit a bit into this group. But I think nevertheless it would be interesting to share some general ideas, especially on plasticity and some methodological things. So I did this bit by bit. Maybe you can put the whole screen on by clicking 2 additional times. Yeah, thank you. So this is the approach that we use in my group. We usually combine fMRI and brain stimulation, be it TDCS or TMS, either in a subsequent or in a simultaneous fashion. And the first approach is the localization approach, where we're targeting functional relevance of task-specific activity. I've done a lot of TMS studies on that, and I think this is a bit boring because usually you only have the behavioral outcome measure. So you first do fMRI to localize activity of interest, and then you target specific nodes within the network. And this only gives you some insight into potential behavioral changes induced mainly by TMS in my case. So that's why I got more excited about the second approach, that is to apply, especially with TMS, plasticity inducing protocols, mainly in inhibition effects. And I'm especially interested in short-term reorganization and adaptive plasticity. So with the idea, what does the brain do when we disrupt a key research? And then I've also talked a bit a concurrent TDCS fMRI study where we were mapping immediate stimulation effects during language. Very fascinating, because we have this opportunity of investigating the direct neural correlates of stimulation effects. I was also always thinking a bit about combining TMS and fMRI in a simultaneous fashion, but I'm a bit too scared about the technical issues. So yeah, next slide, please. So about the first approach, a bit more detailed, because this is the main jobs we're interested in. So the idea is to map rapid short-term reorganization, the immediate response of the brain after a focal challenge. The questions are how a neural networks for a specific cognitive function, in my case mainly language and speech, compensates for a controlled focal perturbation. And I'm also very interested especially in post-stroke aphasia, which makes a nice connection to Paola and also to Jenny. And I think that especially TMS or also cathodal TDCS might be a nice reorganization in the healthy brain. So our approach is to modulate task-related activity, also connectivity, especially effective connectivity with PPI and DCM, and relate this to behavioral changes induced by mainly inhibitory protocols, MVPA, graph theory, and RSA for analysis purposes, effective connectivity, and we're also interested in the underlying structural connectivity. And next, please. Yes, and for the concurrent TDCS and fMRI, as I said, the direct immediate neural correlates of stimulation effects are also of great interest for me to investigate how network activity and connectivity changes during especially a nodal TDCS, and whether we can use this method to increase processing efficiency during acquisition of novel skills and later then also for therapeutic purposes. And here the approach is the same. In the study that I did with Paola, we modulated task-related activity, connectivity, and related this to behavioral changes. And we used the standard TDCS montage with 1 milliamp. We tried 2, but this wasn't possible, as the previous speaker already said. And here the approaches are the same. So yeah, I think I have a final slide on other approaches that we use. So next one, please. Yes, so this is just to tell you what I'm not only interested in these methods, but also in rhythmic stimulation, modulate neural oscillations. Then also we did a bit of simultaneous TMS and EEG to modulate ERPs and neural oscillations during language. I'm also very interested in multifocal TMS approaches and condition and perturb, which means that you do this in a subsequent fashion to study intra and interhemispheric interactions and compensation. So I think that's the realm of what I'm doing and what I want to do. And I'm also now very interested in MR spectroscopy. I don't have any experience with this. So yeah, I think this would be a good starting point for collaborations.
Good. Thank you.
Thank you.
Thank you. And now, Catherine.
Can you hear me?
Yes, loud and clear.
Okay. I just want to make sure. So as I mentioned before, I'm very interested in the role of the cerebellum in cognition, cognitive development, and neurodevelopmental disorders. You can go to the next slide. Can you put to the next slide, please? And so basically, the way that we're using TDCS is as a manipulation and a modulation. So we're trying to investigate what the cerebellum is contributing to cognition. Obviously, it's not the only region of the brain involved in cognition. And so what we're interested in is what happens to behavior, but also to brain activation changes. And so we're interested in both what's happening in resting state, connectivity changes, but also task performance and healthy young adults, but also in clinical populations. And so this is our basic approach. We use the Neurocon system. We also have not been able to get it to 2 milliamps ever. So we have been doing this at 1.5. Our behavioral studies are at 2. We use 5x5 electrodes. We use the saline sponges, and we've been, again, doing this in a 3T Siemens Prisma system with both task and resting state. One of the cool things about doing it in the scanner is you actually get an idea of electrode placement relative to brain anatomy. So on the bottom, you can actually see the electrode on the MPRH scans, and one of my students is very interested in, how accurate are these placements relative to the regions that we want to modulate, given that we're using the Indian as a reference point. We acquire a field map and resting state during TDCS, and they do the TDCS inside the scanner. If you go to the next slide, I'm just showing some of our designs. So we've done a couple of different designs. And Thompson was saying, the idea of a baseline, how do you know what you're referencing to? Our published studies so far have basically used what we call a pre-post design, and had different groups receiving different types of TDCS. So in this case, what happens is they go into the scanner, they get a structural scan, we get 7 minutes of resting state, a task prior to receiving TDCS. They get the TDCS inside the scanner. and then another resting state, and they perform the task again. So when we're doing the analysis, what we're comparing each subject to is their own resting state and their own task activation patterns prior to TDCS. Currently, we're running a study where we're doing a within subjects design. In part, this is because we have a clinical population. We're looking at young adults with autism, and there's a lot of heterogeneity within that population, like with many clinical populations. And so we basically wanted each participant to be their own control, with each person receiving all three modulation conditions. And so they come for 3 sessions. They're 1 week apart. Again, it's a similar protocol in that they get the TDCS within the scanner environment, and then they do a set of tasks after TDCS. And so each participant has an anodal session, a cathodal session, and a sham session. And so I think this is a great idea in terms of, I think together we can probably collectively answer some technical questions as well as, kind of the practical questions of how to do this. I think there's a lot of potential in terms of asking specific questions and potential translational applications. So thank you for organizing this.
Thank you, Catherine. Thank you. Good. Now the next speaker would be Dr. Ines Violante. And Ines, I'm trying to give you, let me just give you the presentation mode. So
that'll be cool, because I was just adding stuff on the slides as people were
making you a presentator. So now you can share your screen.
Oh yeah, I see. Okay, let's see. Can you see this?
Yes, we can. Yes.
Brilliant. Okay. And yeah, I hope it's clear. If it's small, let me know, because now I lost you guys. I can only see my screen. Okay, so thanks, Hamed, and for organizing this. Been really interesting so far, and be really interesting to hear what you guys are doing. So I'm hopefully just to share a bit of some technical considerations when combining tests, so transcranial electrical stimulation with MRI. And I would just like to start by saying what are my interests. So I'm interested in brain networks, I'm interested in dynamics, so I'm interested in systems neuroscience, and I'm mostly using brain stimulation as a way of modulating brain states. And I'm particularly interested in how those different brain states are associated with or support cognitive functions, and look particularly to executive functions and memory. So I just wanted to start by talking a little bit about the challenges with traditional approaches to transcranial electrical stimulation, which you probably, you've been facing. That's why you wanted to change to imaging. But I think it's important that we just in the same, we discuss them anyway. So issues with traditional approaches have to do mostly with the fact that we might have imaging data either being fMRI or EEG, and we are faced with this difficulty that we have to a priori many times in traditional approaches define which region we want to stimulate. If we're talking about TACS, which frequency and phase, if we have more than 2 sets of active electrodes, and here both with any modalities, what intensity are we going to stimulate? And one of the problems with this is that many times, not always, but many times we define this based on a group. It might be our a previous pilot data that we have, but it might also be data that someone else collected, and they are based on group results. And a lot of this traditional approach has involved doing this just behaviorally. So you apply then brain stimulation, hopefully with a set of controls, brain stimulation parameters at a sham or a different frequency when we're talking about TACS. And again, we're looking at group data mostly. And the 2 main challenges for this have to do with the fact that, yes, they have issues with verifiability. So the effects of stimulation on brain function are really hard to verify if we don't have
a multimodal recording technique. So it's hard to know if we actually modulating the networks or the regions that we set up to modulate if we're not able to indeed look at patterns of brain activity. And the other has to do with inter-individual variability. And we know that this is a big problem in brain stimulation research. We've all heard about issues with reproducibility, and we've been touching on the fact that groups tend to choose very different, there's a large parameter space here, right? And we know that participants are very different, so this might be due to individual variations in neurobiology of the brain, but they become even more extreme when we're trying to apply this to disorders like stroke, where you would see lesions in different sides of the brain, or traumatic brain injury, for example, which is some of the disorders that we've been trying to modulate cognitive function using brain stimulation. And regarding this first challenge, it has to do with verifiability. One of the ways that we can approach this is the reason that we're here also today is that we can combine brain simulation with a simultaneous recording technique. And I wanted just to say why magnetic resonance imaging is a good technique. So you've touched on some of these points just to reinforce those. So one have to do with like the absence of complex artifacts. And Duke will talk a little bit with more detail about noise, but I would say that it's definitely much more manageable than with EEG or MEG techniques. The other has to do with the high spatial resolution that we inherently have associated with MRI. So it allows us not only to look at cortical modulations, but also subcortical modulations. And the majority of studies that have combined imaging with brain stimulation have seen that the effects occur in regions that are not underneath the electrodes, and definitely in our studies we see modulations across networks, so brain networks that go beyond cortical regions. So this is an advantage when we use MRI, because we can really look at take advantage of this high spatial resolution to understand better what modulations we might be causing. The other has to do with the additional modalities. So once you have your participant in the scanner, this is quite nice because you can acquire also spectroscopy, you can acquire structural imaging, either being an MP ratio will do anyway, but you can do volumetry on this, but you can also acquire DTI and have information about white matter tracks that might explain some of the variability associated with how participants respond to brain stimulation. And some also cool techniques like current density imaging in the scanner, which allow you to have a current density map, taking advantage of the fact that you have your participant inside the MRI. And there's of course disadvantages to using fMRI. So we know that BOLD is an indirect signal, is a surrogate signal of neuroactivity. And we also know that compared to, so particularly for those interested in brain oscillations, we know that we're talking about a low temporal resolution, right? So we only have really access to slow oscillations here. Although there seems to be some relationship between the envelope of those and the oscillations that we can measure with electrophysiology techniques like EEG and MEG. But what I would say is, like, all in all, I think we have like a still quite positive message. I think MRI can be a quite useful technique to use, and it's great to see that so many of you are interested in doing so. So now I'd just like to move on to some of the technical considerations of when you're doing this inside an MRI scanner. So this setup that I have here is using the Neurocon stimulators. So I know that other manufacturers have come up with different solutions. And I know that, and there's people already mentioned they're using neuroelectrics, and I know there's others using the esoterics, but I'm just going to focus on this because I have much more experience with the neurocon, and I know I've noticed that some of you are using it as well. So in our case, what we have is more than one stimulator, many times for one or more stimulator. And the neurocon comes with this filter box option that you have in the MRI control room. And then you have another filter box, so another radio frequency filter box that you're supposed to put inside the scanner bore. And I'll show you just an image on how this is not such a great solution, even if you put this filter box really close to your penetration panel. And this can explain perhaps some of the technical difficulties that Ben was talking about. So what we did is that we modified this, and we actually have our own RF filter in the penetration panel, which drastically reduce the noise that we are observing, and I have a nice video of how bad that could be. I've also heard of people talking that they couldn't get to 2 milliamps. We certainly can. So Lucia, one of the studies using the neurocon system, so one of the studies she talked about, and I'll just briefly mention it again, did a 2 milliamps, and we also did a 1.8 milliamps in the remote mode. And this is a mode where you actually want to control your stimulators, not manually. They're not very easy to control, the neurocon ones, manually, but you can connect them to digital to analog converter and connect them to a computer and easily control them. And with this, we did for TDCS 1.8. I don't know if some of the issues that you're having perhaps with resistance might be, I've heard, I think Catherine talking about using saline. Don't know if that was the case. So I would say no, just use gel. It's a much better solution than using saline.
Yeah, the reason we started with saline and then didn't want to change halfway through the study, but we're going to move to the gel after. Yeah,
I would actually recommend gel.
Yeah, we had the same problem. We also used the 1020 paste, and we couldn't make it to 2 milliamp because of the impedance. It was always too high. So I would love to hear how to find a solution.
Yeah, absolutely. So then in terms of other technical considerations, I would say safety, right? So thinking about what equipment you can put inside the scanner and how you should position it. And different manufacturers will have specific instructions for their equipment. I'm just going to say in general what would make sense for any sort of equipment. And this is my experience doing EEG fMRI as well. So avoid loops. Avoid long cables. Here, try to avoid anything that goes to a length, anything between a quarter or a half of the wavelength of the magnetic field that you're using. So that's why many manufacturers will give you shorter cables. They typically are less than 50 centimeters or so. And this is not, yeah, I realized neurocon would like us to route the cables to the right side. We typically don't do this. We try to have the cables as medial as possible because this reduces noise and reduces also the amplitude of heating that you can have at the tip of your cable. We definitely burn a couple of resistors. So those cables have resistors, 5 kilo ohm resistors in the tip. And we definitely burn a couple trying with phantoms, different things, particularly if you want to do spectroscopy. So I can advise you guys on that. So this is my next step, which is like which sequences are safe to use. And this is because they are not all the same. So the magnitude of your radio frequency pulses will change depending on the sequence and how fast those are being changed. So manufacturers, again, will have specific instructions for their equipment. And typically, definitely with the neurocon, they say they only try this with MP-Rage and the gradient echo EPI. But you can use other sequences like spectroscopy. So if we've been doing megapress to measure GABA concentration and press sequences, but we had to do some alterations to the typical sequence that we're using without putting the cables inside the scanner. And that's because those tend to be quite SAR intensive sequence, particularly the megapress. And then, yeah, checking your impedances before placing the participant in the scanner and throughout. So typically outside the scanner, I'm sure you do this as well. We check that the impedances are good, the participants are comfortable to go in, they won't just jump if they feel something in their heads. But we also do these checks throughout our first sequences, so localizer, MP range, and we tell our participants that we might do this. So they are aware, and before starting any sequence, we check that our impedances are good, because it's really a faff if you need to go and change, put more paste, or go inside the scanner and need to recalibrate something. So it's good to have it early on. In terms of image quality, I'm not going to touch much on this because I'm sure Duke has an excellent presentation on all types of noise you might have and how to avoid them. But this is an image of our phantom, and you might already see here some noise. I'll just play this video, and you'll see this RF noise in the phantom. So this recontamination was one of the reasons why we created this additional filter in the penetration panel. So once we've done this, we no longer had RF noise in our images. And this is really because the solution of having the RF filter box closer to the waveguide is never as good as having it going through the penetration panel. In terms of design, I was really interested because you already touched on some of the things that I wanted to say. So I think it's considering what you want to optimize your design to. So is it the case that you want to detect behavioral changes, and those might take some time to accumulate, either because the stimulation takes time to accumulate, or because your behavioral task needs a number of trials, right? Otherwise you wouldn't have the power to detect a behavioral change. For those, there's many examples of using both TACS or TDCS throughout the scanner session. So either run or having participants coming in different days. And this is an example, for example, with TDCS that Lucia mentioned, but this is at 2 milliamps, doing simultaneous brain simulation, so TDCS and fMRI for anodal, cathodal, and sham stimulation. Another design that you might think of, and Hamed covered this beautiful, I'm here just talking about simultaneous, I'm not talking about pre and post, is thinking about is having a design that is optimized to detect neurophysiological changes. And for this, we have been using short blocks, which we've find they're quite efficient to detect modulations in bolt signal and to calculate connectivity using PPI analysis, for example. So in this case, I have a block with task, whereby you either have stimulation or sham in the same run, and the same with your rest, so your baseline. So you have a baseline also with stimulation and no stimulation. And this is a study with Lucia. She mentioned it briefly, doing cathodal, sham, and anodal stimulation within the same run, which really allows us to see modulations, neurophysiological modulations due to this different stimulation conditions. In this case, 1.8 milliamps. So yeah, just to conclude, I think using simultaneous TACS in imaging it should be used, and we should be using more of it to address verifiability. I would say this is safe, and the setup is not that complex. So I think and I think that's why this initiative is great, so we can share the solutions that we've come up with. And just to reiterate, the MRI is really a powerful approach. So it will offer you high spatial resolution, and you can use all these complementary modalities that will give you extra information to perhaps explore variability between your participants. And there was a wide range of possibilities for multimodal analysis as well. And yeah, again, might be particularly helpful to understand variability, particularly in clinical populations. And just, yeah, to finalize, I'm really excited about this initiative. I'm open to discuss, collaborate with people, share my data, and I think a multi-center study could be a very interesting way to go. Thank you, guys. I'll try to stop sharing.
Thank you very much, Ines. It was great. And it's quite interesting that I was discussing with Ines in terms of the methods that they are using. We had exactly the same challenge in terms of using the waveguard instead of just kind of the penetration panel. And when we moved to penetration panel, and we basically right now using just the BioPack filters that we have available on our panels. And they are, I mean, they work really well, I mean, compared to the waveguard. So that these are basically the shared experiences. Sometimes when you kind of communicate with companies that making these devices, that would not provide you with all the details. So we can share those things together and see how we might be able to kind of provide a better quality kind of data in the future. And now that the next speaker would be.
Can I add something?
Sure, please.
Yeah. So I heard Ines talking about the going through penetration panel and few other Gesa and Catherine having difficulty with going beyond 1.5 milliampere. And in our study, we are going for 4 milliampere concurrent fMRI and TDCS, and we have been able to do it repeatedly. We have done many sessions now, and there haven't been so many failures. Like and there are failures which I can refer to because there are few things which I learned over the time. One is sponge gets dry and then contacts are lost, and that usually causes in between. So TDCS session is going on and then suddenly it stops. So I think that might be due to sponge getting dry inside the MRI scanner, getting heated up and getting dry, and which increases impedance, and with increase in impedance you lose the contact. So we have been using 1020 paste, and also I talked with Neurocon engineer, and she also suggested to use 1020 paste, which would be better option compared to sponge. But one thing I'm not sure which Neurocon device, if you are using Neurocon device, which Neurocon device you are using, because we are using multi-channel stimulator, and that might be the reason we can go beyond 2 milliampere, but I'm not sure which devices you are using, so that we can discuss about it more later. And going through penetration panel or RF filter panel of MRI has helped us in regard to noise. When we used the waveguide, putting the filter box close to the waveguide, the noise levels were very high, and it was like we were just not satisfied with images that we were able to capture with going through RF panel, going through waveguide. But when we went through RF panel, it really improved image quality. And so yeah, these are the 3 points. But for people who are in EU, the Neurocon FDA equivalent of EU do not allow you to go through RF panel, and you have to go through waveguide. So I don't know how you can get over that problem if you can change your IRB somehow and convince your IRB to go through RF panel. But for people who are not in EU, at least in US it is allowed. So definitely for people who are not in EU, it is an option. For EU, I'm not sure what would be the Gesa, what would be the option. I'm not sure.
Jenny, do you have
these are my
any
points.
notes? Jenny.
Hamid, can I just reply to that point?
Sure,
Sorry, yes, you're exactly right that in Europe it is contraindicated to go through the RF panel, specifically for a safety reason, which is that it's a grounding of the system. So if you put it into the RF panel, then you don't, basically all bets are off. So in our lab, which is the Wellcome Trust Centre of Neuroimaging in London, that is banned. We explored all other options, and I'm happy to say that we came up with a modified system that we could put it through the waveguide that meets the manufacturer's guidelines and minimizes the noise. Because I know Ines told me of her solution, which is what I wanted to try, but it was not allowed in our institute. But we were able to come up with it in an alternative way, which I'm happy to share. And in terms of meeting 2 milliamps, we actually had to work with Neurocon directly to get new filters put into our system. So they manually had to change the system for us to allow us to do that, depending on our MR strength. And we had to give them the exact parameters of our sequences. And actually, it was far from easy. It was actually really technically challenging, and I couldn't have done it either from a safety point of view or technically without physics support. So I think, yeah, different countries probably have different rules, but it is worth flagging up some of these issues. And hopefully that explains potentially some of the discrepancies. Okay, that's enough for me. Thanks.
Good. Thank you, Jenny, for sharing. And now that the next speaker would be Dr. Duke Shereen from City University of New York. And I think that Duke would also kind of share us lots of really great experience that he has in terms of the noise he's facing with TES and fMRI. And then we will have time to discuss together about lots of things that we really like to share together and see how we might be able to help each other. Duke, please. And I'm going to share the kind of give you the, let me just give you the screen. Okay, so now you can share your screen.
All right, can you hear me?
Yes.
And let's see here. Can you see my screen?
Yes, we do.
Okay, perfect. All right. So thank you, Hamid, for inviting me to talk. I think this is a great start to what will hopefully be many more very fruitful talks and collaborations to come. So my name is Duke Shereen, and I'm a research associate professor at the City University of New York, where I direct our MRI facility within the Advanced Science Research Center. So I'm here to talk about noise, specifically TES and MRI noise. So there's, Ines and many of you guys have already touched upon the external noise produced by the setup from the cables, but there's also internal noise from the magnetic fields that are induced in the brain from TES. So I'll talk about both of those sources of noise and how to mitigate them, although we've already touched upon that first main source, and I fully agree with what everyone has said so far. And then in terms of the noise that is induced, that's really noise from the interaction of the magnetic fields that TES creates within the brain while you're trying to use the magnetic fields from the scanner to image. I'm going to focus mainly on TDCS. One, because with TACS, most of that noise, most of that magnetic field sort of cancels out in the time scales that you do your scanning in, and also I have more experience with TDS, so I'll be focusing on that in terms of how to mitigate those noise issues. But back to the external question, that the external noise from the cabling. I'm very interested to hear that in Europe that it's not considered safe to go through the RF panel. I would say I would never want to put any metal through the waveguide, nor anything carrying current, because then you set up basically the potential to create an antenna that can transmit noise, which is, I think, is exactly what Ines showed nicely. So these are two sort of general setups that I think everybody here is familiar with. The one on the left is the setup that Neurocon uses, for example, the standard setup where you have two filter boxes, but your cabling goes through the waveguide. And I think they even ask that you put the cabling all the way around and go through the back of the scanner. We have tested both Neurocon and the esoteric setup, which is, the setup on the left where you go through the waveguide versus the setup on the right where you go through the penetration panel using an RF filter that is the same that BioPack offers. And as Ines has shown, we see that the penetration panel really is the way to go. And had I not heard about the safety issues that you have in Europe with your IRB, I would just say that's what everybody should be doing, but maybe that's something that we can address after this talk. So routing the cables through the RF filter through the penetration panel definitely reduces a lot of this external noise from the cabling. You also want the cabling to go very straight and along the axis of the scanner, which is not always easy depending on your setup. So that's all I'm going to say about the external noise. I think we've talked about it a lot already, and I look forward to talking about it more. But one of the first things that we did when we got our MRI compatible TDCS system was, I'm a skeptic. So they, TDCS modulates brain activity. I have a machine that can measure brain activity. Why not put someone on the scanner and perform simple motor tasks, so finger tapping, with and without TDCS over motor cortex. And so this first image shows without stimulation, and you have very, standard strong signal in the hand motor region. This is a right hand. And then we turn on this stimulation and look, wow, there's a lot more activity, right? There's a lot more, a bigger, more brighter red blob. But I would argue that almost all that increased quote unquote signal is noise. If you look at the bottom left, where and it's basically right underneath where you would place the electrodes. So if you look on the bottom left, there's a very strong area of deactivation that's almost an exact square the size of our electrode on the skull, which is where it was placed. So that for me was a big telltale sign that this is not, genuine activation induced from TDCS. And if you don't believe that, then this is work that was done at City College, also where you see here fMRI activation of TDS versus not TDCS, and it sort of coincides where these electric fields are. By the way, this was done in a cadaver, so we're fairly certain that this is not genuine brain activity. And so that sort of led me to well, what were the sources of this noise? And the first thing to think of would be that, well, the TDCS is creating a current through the brain, and that induces a magnetic field, and that's interfering most likely with the fields that we use to do the imaging and do our signal analysis, especially if we're comparing, signal intensities before, during TDCS stimulation versus off. And so here on the left, this is a very straightforward, functional analysis in a phantom, where I simply looked at the signal intensity, versus a 5-minute on period of TDCS versus off, and there's this area of activation, deactivation, and also in the human, there's just a lot of obvious noise in there. And so the question that I had was, well, how can we go about solving? How can we go about mitigating this noise? And there's 3 approaches. I'm going to touch upon the last 2. That's the area of research that I've been working on. But for using ICA, which has been around for a while, to remove the noisy components, this was actually Ines, who spoke earlier, recently published a paper. And so maybe I won't be talking about that, but perhaps she can address how you can use ICA to sort of clean up the data. But what I will spend the rest of the time talking about are how to use the field maps. I'm glad to hear that many people who've already spoken actually acquire field maps at the beginning of their scans, and there's a way to use that to remove some of these artifacts. And lastly, I'll talk about MICA, multi-echo independent component analysis, which was produced at the NIH with Peter Banantini and his, I think, postdoc at the time, Hardy Kundu, which hasn't really been applied to TDCS concurrent fMRI yet. I think that is very promising. So in terms of a potential solution using field maps, right? So the field maps are readily available on MRI scanners. However, to be able to measure the magnetic field that's induced by TDCS, you would need to acquire at least 2 field maps, one with TDCS on and one with it off. And because that difference will give you the magnetic field induced by TDCS, and it's much smaller than what a regular field map would measure, you have to tweak usually your field map acquisitions to be more sensitive. But if you can do that fairly well, then you'd have you know, a spatially dependent nuance regressor, namely the field map that you can use in your fMRI analysis to regress out that noise. And so I did exactly that in this example of a phantom, the same example I showed you earlier. And in fact, if you look at the middle image there, so the electrodes were placed basically on the equator on the left and on the right. And you can see that the current was not actually going straight left to right, but in this example, for whatever reason, the current was sort of arcing a bit. And the magnetic field was you know, positive on one side of that current and negative on the other. And if you look carefully and compare it to the activation map on the left, I probably should have used the reverse blue-red color coding so it mapped on better, but you can sort of see that this false activation is sort of in the shape of the field map. Now, afterwards, you can use AFNI, I used AFNI, to have the field map as a spatially dependent regressor, and after having done that, you see that it cleans up that noise fairly nicely, and there is no real significant effect of TDCS on versus off in the signal, which you would expect in a phantom. So in the interest of time, I think I'm going to skip a little bit of the background. I would urge anybody who's not familiar with multi-echo independent component analysis for non-bold denoising to read up on some of the literature. I'm surprised it's not used more frequently. I heard someone earlier mentioning their interest in cerebellar functional activity. One great use I've had for multi-echo independent component analysis is to actually be more sensitive to cerebellar activity. But there's a lot of, it's a very generalized method, and it essentially helps you clean up and remove non-bold noise. So I'm going to skip the background here just in the interest of time. But afterwards, if you like, I can go into more detail about how it works. But forget how it works. Let's just go straight to the result. And so I showed you the 2 examples earlier of the phantom and the human. Here in the human on the left, you can see this is the TDCS on versus off at rest. So I just had the participant stare at a fixation cross for about 5 minutes with TDCS on and 5 minutes with TDCS off. Lots of noise. This multi-echo sequence is basically a gradient echo fMRI sequence, but instead of having 1 echo around 30 milliseconds, for every TR it requires a whole volume at, I think this one had 6 echo times, something like 10, 20, 30, 40, 50, 60 millisecond TEs, all at within per TR, right? And so the image on the left, I just used that central echo volume in the time series at around echo time equals 30, and compared before, during, and during TDCS on versus off, and that's the image on the left. The image on the right is the same exact data, but we use those extra echoes to remove any non-mono-exponential decaying signal. We use that as a regressor. It's a very straightforward tool within AFNI. And then once you sort of denoise your raw data, you go about your pre-processing as you normally would, you know, align, et cetera, blur, and then, you know, do your GLM, and you see a lot of the noise is gone. This blob on the right there is, I'm thinking, might be related to sensory activation, right? Because you can feel it when the TDCS is on versus off. This is, by the way, at 2 milliamps stimulation. So just to wrap up there, so you know, design considerations. I know as many of you have talked about it. Maybe I should, you know, we should open up the floor to discussion about sharing our methods to mitigate that. It seems that there, you know, in addition, whether you're going to do concurrent fMRI TDCS versus fMRI before or after TDCS would be one quick way to remove any potential artifacts, right? So you don't have to do concurrent TDCS fMRI. If you want to do it though, I would stress caution and perhaps these examples I gave of using field mapping as a nuanced regressor or multi-echo ICA, those are active areas of research that I am currently working on, and ideally we would do both. So one nice thing about this multi-echo fMRI is that for every TR you can collect data at multiple volumes, multiple echo times. You can use that to actually also make a field map. So my current research project in this direction is to have one sequence that rules them all, so to speak. So one multi-echo fMRI sequence that can do essentially dynamic field mapping every TR and have a dynamic spatially dependent field map as a nuanced regressor in addition to using the multi-echo ICA method. And so with that, I thank you for your time.
Thank you very much, Luke. It was great. So we can see there are basically many areas for collaboration. Now that the panel is open for discussion, so we have another something like 25 minutes or something like, let's say, 20 minutes of kind of potentials for collaboration, and I'm looking forward to see what other people would like to share or discuss or make questions.
May I say something?
Sure, go please.
Okay, so I think your idea of a consensus paper, and maybe that could be a mixture of a consensus and review or meta-analysis, is very good, because I think that would be something that the field definitely needs, so that we're not doing the same mistakes over and over again, and that we first have something like a status quo, that we know what has been done and what the values are. So I would be definitely interested in in writing such a paper together. I think this is a very good idea.
Great.
Hi. Yeah, I had a question about the previous talk, if that's okay.
Yeah.
Hello?
We do not have your video. Can you introduce yourself, or?
I'm Henry Chase at
Okay,
Pittsburg.
Henry,
I don't have a video.
We cannot see your video, but please go ahead.
Hi, sorry. So just a question about the ICA denoising. Obviously ICA relies on sort of spatially structured noise in some way. And often, the typical methods, the fix method, I believe relies on certain sort of characteristic patterns of certain artifacts with certain kinds of quality. Is there any evidence of, I mean, with the noise that we're talking about, are we talking about, what's the structure of it? Is it IID? Is it spatially consistent, or is it sort of a burst of white noise that's sort of occurring randomly? If so, if we have a sense of what those characteristics of the noise are, might we optimize our pipelines to try to address that? And just to follow on from that, this is another point where the collaborative effort might be useful insofar as we might be able to share these sort of characteristic noise patterns and these kind of methods as well.
So I think we shouldn't conflate the two different types of ways to use ICA to clean up the data. I mentioned in the beginning that I think, Henry, the sort of ICA that you're referring to is the sort of methods that Ines has recently published in her paper on TDCS and REST-TDCA fMRI, where she uses melodic denoising. So maybe, Ines, you'd want to speak to his point, and I can follow up with the multi-echo ICA if that's.
Yeah, so sure. We use the fix melodic from the FSL toolbox that you're mentioning. To be fair, most of the noises that we had before were like RF introduced noise. I haven't seen like a particular pattern of noise that I can described once we got rid of the RF noise. So it'll be interesting to see. And there's nothing in particular in fixed, because we went through all the data that we can see is a particular component associated with noise. And we try this in phantoms, and we follow up this with humans. So we went from no RF, we went increasing the RF powers. So I don't know. I don't know if you saw a particular type of noise that you are keen in discussing.
No. I haven't seen anything. It's just we have a sort of denoising method that we have, and it will be differentially susceptible to high frequency, low frequency, spatially. You know, if it has a particular spatial structure or not, it might be more or less sensitive to that. I mean, these are considerations. You know, is it happening at a lower frequency? Is it happening? You know, in which case would just a simple high pass filter remove some of it, or would it accentuate it? Things like that. I mean, I haven't looked at this specifically. It's something maybe we could look at. But I just was wondering if you had any pointers on this, because and as you say, I think I believe with fix there are certain templates and certain patterns which, you know, I believe that the method looks for. Insert, you know, typically it's looking for physiological noise which has a certain distribution, maybe in ventricles, maybe, you know, and maybe towards the periphery of the brain is often high, you know, has a high standard deviation in terms of the voxels. So, you know, so we have certain priors, we have certain expectations about where the noise is, and I think, you know, I'm just wondering if that can be leveraged, you know, in terms of TDCS, concurrent TDCS, if there are certain characteristics we can look out for in terms of where the artifacts are and how we might correct for them.
Absolutely, because with fix gives you also the possibility of training your data. So what we did is that we trained our data in a particular data set we acquired in a lab that you can then apply, and that gives you at least an idea of how the pattern of your noise, both temporally and spatially, both if it's either high frequencies, low frequency, and if it's localized in a particular region. I don't think we've identified something that is a clear pattern of a noise like Duke has shown, but again, what we did was a simple sort of quality control with phantoms, and then a pilot study really looking, so no RF on, and ramping it progressively and doing on and off, doing the same acquisition and calculating standard deviations and seeing if there's a particular difference or not. The field maps are actually great because as Duke was mentioning, and we did acquire them at the beginning, and now we just realized, so Lucia was just telling me that we do have them on and off in all participants because of the multi-echo. So again, it'd be cool to look at it. Yeah.
Also, I chime in here quickly. It'd be interesting if maybe Henry could start to try to identify what the source of the noise is, right? So I mentioned how there's these, we've all talked about how there's these external noise contributions and also the internal noise from the field induced in the brain. I'm curious, do you have RF filters in your setup, Henry? Going through the penetration panel, or are you going through the waveguide?
Yeah, I realized your talk, I need to check back on that. We do have a filter, a biopack filter that we're using, but I need to check up on what we're doing. We did some very sort of peripheral checks of noise, and we haven't really seen anything, which is why I've been asking. We haven't seen much in the way of distortion of our images or anything like that. So yeah, so your talk has sort of given me a load of questions to go back and check up on.
Good, great. Do we have any other question? Now we have others as well. We have Adam Woods here, who is really kind of well experienced in terms of putting together these sorts of consensus papers and methodological notes. So we definitely need to have his input as well in terms of how we might be able to move forward and think about something like a consensus paper. Adam, do you want to kind of add any notes to that?
Sure. No, I think the, can you guys hear me okay?
Yes.
I came in late. Sorry, I was sitting in a very awful dissertation proposal for the last 2 hours. So I apologize for my tardiness. But yeah, I mean, I think a consensus paper in this domain, there's really so many elements to this that could make for, I think, great consensus papers, whether it's a giant singular consensus paper or a well-planned couple or three, we can discuss. But ultimately, I think focusing in on what we want to achieve in a consensus paper, and then we start from there with circulating a rough outline and filling that in, assigning, you know, the different sections and or groups to certain sections to work kind of offline from the main group with a series of deadlines to pull us together. But all of those things are definitely doable, and I think we have a great group here for these types of consensus papers.
Good, good. Ben, do you have a question and any note to add?
Well, excuse me. Yeah, it's a little bit on a different topic, but working in visual cortex, I'm always extremely jealous of people that work over motor cortex who have this really nice objective MEP output measure and kind of know what the brain stimulation is doing. We never know really when we move to a different cortical area if it's doing the same thing. And so with imaging, we can look at functional changes, we can look at spectroscopy, but with all the imaging experts here, I was wondering what else can we do with imaging to tell us how the brain stimulation is affecting, or what the mechanism of the brain stimulation is over non-motor areas. Are there techniques that we could, like emerging techniques we could use to address those questions?
Well, I'll comment on some of the work we're doing. It's not necessarily a measure of change in physiology per se, but we've got an R01 we're in the second year of now from the Brain Initiative from NIH, where we're using MREIT, magnetic resonance electrical impedance tomography, to look at an objective metric of current flow in the MR environment. And that's really kind of leveraging the artifacts we kind of heard about here, using a specific approach of TACS to perturb the signal in a controlled fashion. And the physics are far above my pay grade by all means, but from this team here at University of Florida and Arizona State, we're using that approach to actually map the current flow versus predictive models, which are great, but we're going to use that data to not only improve the predictive models, but also to directly query what elements of the current flow are contributing in the context of a working memory paradigm we're doing with TDCS and these same participants to perturb connectivity and you know, particularly in frontal and parietal regions, as well as to change bulb response and look at, okay, what elements of the current flow are actually contributing to this? Is it local change? Is it remote change? What have you? And this is really an emerging technique that a handful of groups are working on. I think that'll help us in some ways, where we have this additional piece of information of knowing how much current went where, voxel to voxel. That's not the final answer for all of this, but I think as that technique becomes more and more available, and we're really working on currently deploying this for both Philips and Siemens scanners, that will help in part of this. But I think your question is absolutely a good one. What other techniques in terms of multimodal imaging can we use? Not, you know, and it's not just fMRI, it's not just MRS, but there's also ASL and a number of other techniques that can be useful in this.
Any other comments from other colleagues?
Yeah, I'd quickly like to comment on the idea of sorry, multi-lab study that came up a few times. So what might be relevant is that we are currently running a multi-lab study for TACS, and I guess the outcome of that multi-lab study would be quite relevant for the purpose here, right? Because we do not only have to understand how TES fMRI works, but also how TES in general works, right? So we had a meeting in December, and now our second meeting will be in April, quite soon, in 2 weeks or so. So if you're interested, just send me an email and I'll put you on the email list. The meeting will be live streamed, so everyone can attend and you can send us questions.
Good. Any other comment, idea, question that people would like to share?
Hello? Hello? Can I see you?
Yes,
Okay.
yes.
Okay. I have very quick comments to the TDCS because during when I do the studies, we found and researched the literature and found, for example, is treatment the addiction. We can most paper we use to stimulate the DRPFC, but which side of DRPFC is really inconsistent to each other. Some paper found use the left side, some paper use the right side. So I think this is very maybe interesting, maybe very difficult things. Which student point is worked? I think this is very, very important, and I suggest maybe we can during our paper, and we can to give some discussion of this one, and it should be very important issues for the TDCS in the future, especially in the clinical application. Okay, so that.
And I think part of what makes that so complex are what we have seen from some motor physiology MEP studies. We see that the intensity of stimulation and duration of stimulation will actually alter, at least in terms of motor cortex response, whether you're seeing excitability increase under the anode or excitability decrease under the cathode. You take it to 2 milliamps, and that data suggests from a series of relatively recent papers that you get net excitability under both. Now, it's a huge assumption that the frontal cortices are behaving the same way as motor cortices. If that assumption were true, it could be demonstrated with a number of these imaging methods like fMRI and otherwise, looking at change relative to different dose parameters. It may be that we find that, you know what, you don't have to necessarily pick, if you're going for excitability, 2 milliamps, F3, F4, you're net increasing, but we're still not there yet in terms of understanding, which is a point someone else was making, that, you know, it's great when people are working in the motor cortex, you got this great outcome. But I do think that with imaging, fMRI, and other methods, especially looking at GABA concentrations, GLX concentrations, and these various shifts, we can start to, I think, understand what these different dosing parameters are doing in different regions in the brain. And so, yeah, I think you bring up a really hot topic that presently a lot of people are asking questions about.
I completely agree with that, and I think that when I'm picking parameters for experiments, I'm kind of stumbling in the dark a little bit, and so I kind of go for the strongest one that I can do because that feels like it should be a good idea. But then I was just reading yesterday in the TRNS literature, it's an inverted U-shaped function depending in that theory how much noise you inject. So the idea of having some data, some region-specific data on dosing is an excellent one, and one that I thoroughly support.
Another thing that could be of interest too is individual subject data. So not everyone shows the same directional response to TDCS. And if we're ever going to, you know, a lot of us have interest in clinical populations. If it's going to be a treatment protocol, it would be great to actually be able to use the imaging if there's a relationship between sort of the direction of the response and what their brain looks like in a resting state prior to TDCS. It might be very important clinically, both in terms of, you know, localization of electrodes, but also in terms of, you know, what you would predict in an individual patient, how they would respond. So I think that in the data we're all collecting, there's also the ability to ask that question in a way that maybe if we pull some data, even though there's a lot of different protocols, but we could look at the individual subject differences, and it could be really informative.
I think that's a great point. And one of the things we've actually worked on here, we have a phase 3 TDCS trial ongoing, and we're getting imaging pre-post and 1 year. But we've actually taken the new released ROAST protocol for computational modeling, and one of my postdocs is specialized in this area. And we've, in our supercomputer, actually managed to get this working with our infrastructure to massively parallelize individual processing from all those T1s. And part of the idea behind that is in all of our studies we've collected with in-scanner TDCS, but also in these treatment paradigms, we're going back and modeling every individual and looking at what elements of individual variability in brain structure and potential conductivity based on those variations are potentially contributing to the differences in response, but then trying to take that further and integrate it further with our fMRI data and change data and other modalities. And I mean, in any of these studies where we have T1s, which we all do if we're doing, you know, imaging with TDCS, and more and more trials are having this as well, the pulling of that data and using these more automated processes for extraction of relevant data could be a really cool and groundbreaking approach, especially if there's information on how behavior or whatever outcome measure changed as a result of that. I think it's a wonderful idea.
Hey all, sorry, this is Ben Hamstead from University of Michigan. I'm just jumping in to follow up on those last couple of points. So, you know, I'm actually doing a lot of work now with the aging in various types of dementia. And so, you know, we're just starting up an R01. Adam's a consultant on it, with where the goal is to enroll 140 MCI patients and look at varying doses from 1 to 3 milliamps plus a sham. We'll also get amyloid and tau on all of these patients, so we'll actually be able to look at disease burden as opposed to just risk. You know, and so I've talked to the program officer, and so I'm going to put in a supplement to be able to use that ROAST modeling. So again, we can look at these individualized factors, and some of the preliminary data that I've worked with Abi Data from Soterix on, you know, in just a small group of 12 cognitively intact older adults, we showed literally 100% variability in how much current was delivered to the targeted brain area, which, you know, is fascinating because if we're talking about, you know, the stimulators only being able to get up to say 1.5 milliamps, those data were all at 2 milliamps. And so it suggests that that reference person would need 4 milliamps to get the same as the person who was the highest. So, you know, I think that those factors are all going to be increasingly important to look at. And I personally, I think looking at these scans of the patients that I'm working with day in and day out, there's so much atrophy that I don't know how well the healthy 30-something-year-old model that we're all accustomed to looking at actually represents these patients. And then if we factor in the amyloid and tau or other disease pathology, it's kind of the Wild West, I think. So, you know, I'd be happy to kind of open up these data for any of these purposes. We're also getting a lot of patients with PPA in there. So I know a couple of people mentioned interest in language processing, which could be interesting to look at as a comparative to, say, more traditional stroke populations. So anyway, I think this is great.
Good. So that's great. That's kind of the notion of thinking about how we might be able to have individualized protocols, having ROS or SIMNIBs or other protocols that we have, combining that with fMRI. And you know that there are just really few studies that they try to do a combination of those types of computational head models and fMRI in the group level. And that is another kind of challenging issue that we probably kind of in the next webinar, we will be able to discuss about that as well, in terms of how we might be able to bring kind of group level analysis on an fMRI and combining that with ROST and SIMNIBs. And there are some kind of breakthrough kind of developments in that area as well. So that will be something that we probably can discuss. And we have done quite interesting things for that with colleagues like Duke and kind of Marom, Bikson. So there are things that we probably can discuss about those things as well. So, okay, we just have 2 minutes, just kind of a few last minor notes. First of all, the second webinar is going to be in 3 months. So it would be June 27th at the same time on Thursday. So it would be 10 a.m. to 12 Eastern time. So hopefully by the 3 months, we will be able to move forward and do a few things together. So what we are going to do, the next step, those who are interested to actively contribute on that consensus paper, please send me an email, because we usually have 2 groups of people. We have a group of people who basically actively contribute and write a lot in the consensus paper, and there are others who would make comments, and there is another third group that would basically endorse at the end of the day. So that's kind of the combination that we probably have at the end of the day. So those who are going to be lead authors and active contributors, please send me an email in terms of if you are kind of interested and what are the areas that you're basically more interested to write about in the consensus paper, to let us kind of move forward and put a kind of an outline together and discuss how we might be able to move forward for that. And then those who like to share any specific sort of experience in terms of even a specific type of data that they want to share, or a specific sort of experience that they like to share, or there are potential collaborations. So those things that we really like to share in the email list that we already have, that would be something like 200 people who are basically doing these kind of areas of activities in the field. And the fourth item would be, I've been discussing with some of the program directors in different NIH programs, and it seems that we might kind of have potentials for having a joint collaborative sort of kind of proposal together in the future. But if you are informed about any potential granting opportunity that would welcome joint collaborative data collection in different sites, that would be something that we probably can discuss together in the future as well. So it's right at the time. So it was great to have you all in the meeting. We had 36 participants from across the board, and that was great to have you all together today here. And I hope that we will move forward together to do something that will be invaluable in the future. So have a super nice day, and we will keep in touch.
Thanks for organizing.
Thank you.
Thank you.
Thank you.
Bye, everyone.
Bye.